Hello everyone. The next topic which we will be discussing is about the structure of protein. So the first question that arises in our mind before we study the structure of protein is that what makes up the protein structure? So the answer to this question is a protein primary structure refers to the amino acid sequences in a polypeptide chain. So they are peptide bonds that are made during the protein biosynthesis process that holds the primary structure together. Now, the next question is what are the protein structures and what are the stages of protein structure? So there are four stages of protein structure. There are primary, secondary, tertiary and quaternary. So these are the four levels of protein structure. Now, before starting with the structure of proteins, we will first discuss what are proteins. So proteins, they are large molecules that are composed of basic units that is known as the amino acids. In addition to containing carbon, hydrogen and oxygen amino acids, they contain nitrogen also. Protein structures, they are very large and complex molecules and they are formed by one or more twisted and folded strands of amino acids. They are highly complex molecules and they are actively involved in the most basic and important aspects of life. So these will include metabolism, movement, defense, cellular communication and the molecular recognition. So this is the basic terms that what are proteins. And the proteins, they're essential for the growth and development of an individual. If we take the examples of protein, that what are the food items that contain protein? So they are eggs, pulses, milk and other milk products that form the major high protein foods for the body. So now, what are the structures of protein? There are four types of structures of protein, that is the primary, secondary, tertiary and quaternary. So we will be discussing each of these structure of proteins in detail. So first we will start with the primary structure of the protein. So this is a diagram which is depicting all the four structures of proteins that are being depicted here. Now the primary structure. So the primary structure of the protein is nothing but a sequence of amino acids in a polypeptide, in a linear polypeptide chain. And they are formed by the amino acids in a particular sequence. So they are formed by the amino acids in a particular sequence. Changing the position of even a single amino acid will result in a different chain and hence a different protein. The primary structure, it is held together by peptide bonds and that are made during the process of protein biosynthesis. It is determined by gene corresponding to the protein. So changing the position of a single amino acid will result in a different chain and hence a different protein is being formed. So this is what the primary structure of the protein is. Now if we look for more detail, then the example here is the hormone insulin. It has basically two polypeptide chains, that is A and B. It will be shown in the next diagram that there are. So this is the example of the insulin chain. So there are two chains which are being depicted here, the A chain and the B chain. So what is happening is each chain has its own set of amino acids that is assembled in a particular order. For instance, this sequence of A chain starts with glycine. So this starts with the glycine and at the N terminus and the asparagine with the C terminus. And if we see the chain B, it has a very different sequence. So the sequence of a protein, it is determined by the DNA of the gene that encodes the protein. A change in the gene sequence will lead to a change in the amino acid sequence of the protein. So even if we change one amino acid in a protein sequence, it will affect the overall structure of the protein. So if one of the amino acid is being replaced from here, then the whole protein structure will change and it will also affect the function of the protein. For example, if we take the example of a disease, which is a sickle cell anemia, which is an inherited disease that affects the red blood cells. So what is happening in that disease is one of the polypeptide chains that make up the hemoglobin, the protein that carries the oxygen in the blood, they have a slight sequence change. So the glutamic acid that is normally at the sixth position of the hemoglobin beta chain, it has been replaced by the valine. So this is how a change in the sequence will cause the sickle cell anemia. So this is the example here. The next we will talk about is the secondary structure of the protein. So what is the secondary structure of the protein? The protein structure that is formed by folding and twisting of amino acid chain, and it refers to highly regular local substructures on the actual polypeptide backbone. So the secondary structure, they have the alpha helix and the beta pleated sheets, and it was being suggested by in 1951 by Lingus Pauling and other scientists. So the proteins, they do not exist in just simple chains like that of the primary structure. These polypeptide chains, they usually fold due to the interaction between the amine and the carboxylic group of the peptide link. So this structure refers to the shape in which a long polypeptide chain can exist. They are found in two forms, that is the alpha helix and the beta pleated sheets. This structure arises due to the regular folding of the backbone of the polypeptide chain due to hydrogen bonding between the CO group and the NH group of the peptide bond. So if we talk about the two helix here, so there is a alpha helix and the beta pleated sheet. So what is alpha helix? Alpha helix is one of the most common ways in which a polypeptide chain forms all the possible hydrogen bonds. By twisting into right-handed screw with the NH group of each amino acid residue hydrogen bonded of the CO of the adjacent turn of the helix. So these are basically twisted in the right-handed. These are basically twisted in the right-handed screw. Next is about the beta pleated sheets. So beta pleated sheets, in an arrangement, the polypeptide chains, they are stretched out beside one another and then bounded by the intermolecular H bonds. So in this structure, all the polypeptide chains, they are stretched out to nearly maximum extension of maximum extension and then laid side by side, which held together by the intermolecular hydrogen bonds. So this is what is known as the tertiary secondary protein structure. So they are formed by hydrogen bonding in the polypeptide chain. They have two different conformations, that is the alpha helix and beta pleated. And the alpha helix is a single spiral. It is formed by hydrogen bonding at every fourth amino acid. And the beta sheets, they are formed by hydrogen bonding between two or more adjacent polypeptide chains. So this is about the secondary structure of the protein. So next we will talk about this, the tertiary structure of the protein. So the tertiary structure, it is a protein structure which is formed when there are twists and fold of the secondary structure again, and they form a larger three-dimensional structure. It may include one or several domains. The alpha helix and beta pleats are folded in a compact globular structure. So the tertiary structure, it is the final three-dimensional shape that has been acquired by the polypeptide chains under the attractive and repulsive forces of the different R groups of each amino acid. And this is a coiled structure. It is very necessary for the protein functions. So this is what the tertiary structure is. Now the quaternary structure. So the quaternary structure, it is, what is it? There are some proteins that are made up of multiple polypeptide chains, and it is known as the subunits. So they are known as the subunits, and these subunits, when they together come, they will give rise to the quaternary structure. The protein structure is a protein consisting of more than one folded amino acid chain. And it is a three-dimensional structure which is consisting of aggregation of two or more polypeptide chains that are operate as a single functional unit. So if we talk about quaternary structure, then this structure is exhibited only by those proteins which have multiple polypeptide chains and they combine to form a large complex. And the individual chains, they are called as the subunits. So the exact amino acid sequence of each protein derives it to fold into a unique and biologically active three-dimensional fold, which is known as the tertiary structure. When the proteins, they consist of different combinations of secondary elements, some of which are simple while some other are complex. So the part of the protein chain which have the three-dimensional fold, they can be attributed by some function which are called as domains, and these are considered today as the evolutionary and the functional building blocks of the proteins. So many proteins, uh, most of which are enzymes, they contain organic or elemental compounds needed for their activity and stability. So the study of protein evolution not only gives the structural insights but also connects proteins quite different parts of the metabolism. So this is what the quaternary structure of the protein. So this is the summary, or we can say this is the summary that what we have studied till now. So there is a protein structure which is just a sequence of amino acids. So this is a sequence of amino acid. This is known as the protein structure. Then comes the secondary protein structure which have the alpha sheets and the beta pleated sheets as well. So there are alpha helix and there are beta pleated sheets. So there are hydrogen bonding of the peptide backbone and it causes the amino acids to fold into a repeating pattern. Tertiary protein structure, they are what is seen as the three-dimensional folding due to side chain interaction. So this is the tertiary structure. Quaternary structure, it is protein which is consisting of one or more than one acid chain. So quaternary structure is nothing but the next step between the two or more polypeptide chains, which is from the tertiary structure and is a specific special arrangements and the interaction. So this is all about the structure of the protein. Now these are all the references. Thank you.